Welcome back, everyone. We're now going to move on and we're going to talk about reversible and irreversible injuries, and we're going to discuss apoptosis and necrosis in particular. And we'll start here with reversible, and you can see it over here via this blue curve here. With a reversible cellular injury, the stimulus ceases and the cell returns to normal. So you can see there the removal of the stimulus, whether that's hypoxia, whether it's some chemical agent or bacterial toxin or whatever it is, the cell will return to normal. Now, it is still characterized with this cellular swelling that we talked about earlier, that infiltration of water. And if enough cells sort of swell, then the whole organ is going to sort of stretch or become distended and can turn a paler color. But as long as the stimulus is removed, that cell then, can revert to normal functioning. It's also characterized by lipid accumulation, and we see this particularly, say, in hepatocytes in the liver, when somebody has fatty liver disease and that liver turns quite greasy and yellow in color. However, with an irreversible injury, the stimulus continues and a critical point is reached, or a point of no return is reached, and that cell then will die. Now, there are two processes that lead to cell death, but they are very different from each other. The first one is apoptosis, which is programmed cell death, and this can be physiological and it can also be pathological. An example of physiological apoptosis is in the central nervous system of children, and we're actually born with way more neurons than we need, and some of these undergo apoptosis early in childhood. I would beg to differ with that, because at my age, I think I need some of my neurons back. Pathological apoptosis, then, an example would be, hepatocytes in the liver undergoing programmed cell death when they've been infected with, say, a virus such as hepatitis. Now, the really important thing to sort of get from apoptosis is that it doesn't induce inflammation, whereas the other type of cell death, which is necrosis, does induce inflammation. Now, this little figure over here is actually showing you necrosis. We're going to go through apoptosis and necrosis, which lead to cell death. But again, as I've mentioned, the way they get there is very different. So if you look here, we have our normal cell. There's some sort of injury that's leading to this swelling that's reversible up to a point. Say it's hypoxia causing it to swell. Once that hypoxia is sort of resolved, that cell returns to normal. However, if it's not, then you can see this cell here is reaching this point of no return, becomes an irreversible injury, and that cell dies via necrosis, which does involve the induction of inflammation. And this little figure is just kind of going through this in the same way. We have a normal cell, it undergoes some kind of stress, it's able to adapt to that stress to a certain point, but then it reaches a point where it can no longer adapt, and that causes a cell injury. Or you might have a normal cell that sort of undergoes kind of a direct sort of toxic sort of injurious stimulus that leads to that cell injury. Might just be mild, reversible injury, and as long as there's some sort of intervention or removal of a stimulus, then that cell recovers, right? However, in some situations, the cell injury is more severe, or, it progresses and it becomes that irreversible injury when it reaches that point of no return. And that cell then will either die via necrosis or via apoptosis, which we're going to go through now. Now, just a quick reminder of the manifestations of cellular injury. They do involve these cellular accumulations or infiltrations that may or may not be toxic to the cell. So water, lipids and carbohydrates, glycogen and proteins. So we'll start with the process of apoptosis, which I'm sure many of you have already heard of. So this is an active process of cellular self-destruction, and we call it programmed cell death, right? It involves the activation of a specific set of genes and enzymes. So it's a very neat and ordered process. As I mentioned previously, there is physiological apoptosis, which occurs during the development of body tissue, for example, the central nervous system in kids, and pathological then, where liver cells or hepatocytes undergo apoptosis when infected with hepatitis B. So if you look at this little schematic here, these are the steps of apoptosis. I think to anybody's eye, you can see it does look very ordered. We have sort of condensation of nuclear material here. We get these distinct sort of bulges in the plasma membrane that break off. Then we get this nice ordered cellular fragmentation and the generation of these apoptotic bodies. And then we get a phagocyte coming in there and, phagocytosing these apoptotic cells and fragments and cleaning up the area. And you can see that there's no induction of inflammation. On the other hand, now let's look at necrosis. And necrosis is the result of a sum of cellular changes after local cell death and the process of cellular autodigestion or autolysis. It does consist of phases as well. So there's this early pycnosis phase and this later karyolysis. Okay, so here we have our normal cell. And then we have this injured cell here. You can see we got this general swelling of the cell, including, swelling of cellular organelles. And you can see here that we've got this clumping of this nuclear material. So this chromatin and general sort of clumping of the nucleus. The later phase then is karyolysis. So you remember that prefix kary always relates to the nucleus. So here there's complete sort of nuclear dissolution and also rupture of the cell membrane. And obviously, if you've got nuclear dissolution, you're going to have chromatin lysis as well. So if we put both of these schematics together, you can see we've got necrosis here on the left and apoptosis here on the right. And even just to the naked eye, you can see that this sort of process of necrosis is much messier to this apoptosis here. So we'll just go through this one. We've got our normal cell that gets injured, and to a point, it's a reversible injury and it may recover, or it may then, go on to irreversible sort of injury. And we get all of these steps here of, pycnosis and then karyolysis. And you can see very clearly that this involves inflammation, whereas over here, apoptosis does not. We're going to take a brief look at the different types of necrosis that can occur in different organs and tissues. And these are coagulative necrosis, liquefactive necrosis, caseous necrosis, and fat necrosis. There is another type of necrosis that's sort of indicative of sort of larger areas of tissue death, and this is called gangrenous necrosis, and we will talk about that too. Now, the first type here is coagulative necrosis, which can occur in sort of, lots of different organs, but the kidneys, the heart, and the adrenal glands are particularly susceptible to it. And it's caused by hypoxia as a result of ischemia, and it involves protein maturation. So with this type of necrosis, it involves sort of denaturation of the protein albumin, and that sort of causes it to change from its sort of gelatinous, transparent sort of state to sort of firm, opaque state. And that's due to that cell death. The second type then is liquefactive necrosis. And this can occur due to ischemic injury to nerve cells in the brain. And dead brain cells are affected by this type of necrosis because they have these really high levels of digestive enzymes. And the brain itself has only small amounts of connective tissue. So what it kind of involves is, cells are kind of digested by their own enzymes, and that tissue then becomes soft and watery, and it becomes walled off from healthy tissue, which is what you're looking at here. Now, caseous necrosis can occur in lung tissue as a result of an infection. So, for example, TB. We'll talk about that when we get to the respiratory topic. It's a combination of coagulative and liquefactive necrosis. So these dead cells do disintegrate, but the debris doesn't get completely digested by enzymes. So what you end up with are these regions of tissue that sort of resemble cheese. And that's why it's got its name caseous. And so you can see that the dead tissue there kind of looks soft and granular, a little bit like cheese. Fat necrosis then can occur in the pancreas and other abdominal organs, and it's due to the action of lipases. Remember, lipases are enzymes that break down triglycerides, and once that happens, then they combine with ions such as calcium, magnesium, or sodium ions through the process of saponification, then creating these soaps. And so what you end up with here is this tissue that's sort of opaque, this dead tissue that's sort of opaque and chalky white. And finally, just looking at gangrenous necrosis, which describes death of tissue from severe hypoxic injury and then bacterial invasion. There are two types, dry and wet gangrene. And here on the right, what you're looking at is dry gangrene, which is often, the result of blockages in arteries, supplying the lower leg, and then invasion of bacteria. And what you can see here is the skin becoming very dry and wrinkly, and the color obviously is changing very clearly there from brown to black. With wet gangrene, this can occur in internal organs. It involves white blood cells such as neutrophils that invade the site, causing liquefactive necrosis. And this causes, the organ to sort of become cold and swollen and black. And, because of sort of the bacterial invasion, then this also produces a very foul odor. And of course, if this, infection then becomes systemic, that is going to be correlated with increased risk of mortality. We're going to finish up here with aging and altered cellular function. Now, over the period of our lives, we are exposed to injurious agents as we age, and they include things like UV light, temperature changes, infectious agents, and of course those free radicals that we talked about earlier on, which changed, the structure and function of cells. Now, it's really difficult kind of to distinguish between sort of the normal sort of aging process, which is unavoidable, and we all have to go through it, and sort of distinguishing between that and disease is really hard to do. So the easiest way is to give you an example. So say brain atrophy. Now, during the normal aging process, the brain does undergo atrophy, right? But when there is a disease state, such as, Alzheimer's or something like that, there is way more atrophy. So a significantly higher level of atrophy in that diseased state. Similarly, you could talk about arteriosclerosis. There is, a gradual narrowing or hardening of the arteries as we age. But then when you think about that specific type of arteriosclerosis being atherosclerosis, which involves those sort of lipid deposits on arterial walls, that's when it becomes, diseased rather than just, natural aging. And there are, of course, genetic factors and environmental factors that are involved in the aging process as well. With genetic factors, as we age, our DNA, we get sort of DNA repair defects. And when that occurs, then an accumulation of mutations, which contributes to cellular aging. We also, as we age, sort of accumulate genetic abnormalities, and that then will lead to abnormal cellular signaling. And again, both of these things will sort of cause reduced ability to produce new cells and therefore cause cellular aging. And environmental factors, feed into all of this. And a lot of these environmental factors are on the previous slide here. So, exposure to UV light over a period of time, temperature changes, infectious agents, and accumulation of those free radicals. So Both of these, so free radical damage and reduced enzyme activity, will cause accumulation of damaged cellular proteins and therefore organelles, and therefore, reduced ability to produce new cells. So it is sort of this combination of genetic factors and environmental factors that contribute to cellular aging. All right, and our final thing to look at is somatic death. So we're talking about death of an entire person. And there are these characteristic, changes that occur once a person dies. So these post-mortem changes. Obviously, the person is going to stop breathing. There's also going to be a cessation of blood flow because there's no heartbeat, right? And then the body sort of undergoes this or goes into algor mortis. And this comes from the Latin mortis, obviously meaning death, and algor meaning coolness. So these are changes in body temperature that occur after somebody dies. It's sort of gradual to begin with, so there's a decrease in body temperature gradually, but about 24 hours post-mortem, the deceased body sort of has assumed the temperature of the surrounding environment. The pupils become dilated and they don't react to light, and the surface of the skin becomes pale and yellowish. And there are these sort of really characteristic changes in facial features where the cheeks and the nose kind of assume sort of a peaked effect. And that's because, of the draining of blood from the face and it accumulating in lower tissues due to gravity. Rigor mortis, so the stiffness of death, then sort of occurs within 6 hours after death, and it affects the whole body within 12 to 14 hours. And the muscles do stiffen due to the breakdown of carbohydrate and obviously depletion of ATP. And this gradually then diminishes and the body then becomes flaccid somewhere between 36 and 62 hours after death.